pollution is a side effect of rapid industrialization. Lead or plumbum is one of the most frequently found heavy metal in polluted environments. Phytoremediation is an emerging and low-cost technology that utilizes plants to remove, transform, or stabilize contaminated water or soil. Hyperaccumulators are metal-tolerant plants able to grow on heavy metal-contaminated soils and accumulate high amounts of them. However, they have small biomass and are slow-growing. A possible solution to this is to incorporate metal-accumulating genes into non-accumulating plants. The aim of this research was to perform the first step in engineering metal tolerance in selected wild plant species. Nicotiana glauca, R. graham, is a fast-growing, high biomass and tolerant to white gray jaw condition. This involves a transformation of the plant with wheat phytokeratin synthase gene, subsequently determination of their heavy metal accumulation and tolerance, specifically plumbum and cadmium.
For the methodology, it was divided into two different parts. The first part is focusing on the agrobacterium strain, plants transformation, and culture conditions. For the second part, plant growth and tolerance experiments was highlighted. In this part, new plant gene was constructed and electroporated into agrobacterium tumefission strain. There are two experiments involved, whereby experiment 1 was observing the plant growth and tolerance on the agar medium, whereas experiment 2 observed the growth and tolerance of major plants in contaminated soil. The contaminated soil is a calcareous sandy loam with the presence of lead and zinc concentrations.
First, selection of a natural plant species for phytoremediation. Nicotiana glauca, also known as shrub tobacco, were chosen for GM plant as it fills all requirements. Insertion was tested on Saccharomyces cerevisiae and confirmed through genomic PCR, Southern and Northern blot analysis. Other than that, the APCSI gene were known has ability to increase lead uptake and accumulation. Real outcomes were observed on how this plant handled the lead uptake and tolerance on cadmium. The figure shown the root length of modified and white type plant that grown for 10 days in agar media containing no added lead, which is the upper line, and with added lead, the lower line. The root growth of GM plant was increased by 160%. This shown that the plant able to increase the lead uptake and accumulation. Compared to wild type, the root was seen shorter than GM plant.
So for the discussion, this experiment actually tests on other plant species such as terrestrial and also alezum. So from what they found that they have important limitations, such as this species have specific for one particular metal, so they absorb only one type of metal, and then they adapt to precise climate and soil condition, which means they are not really transferable, and then they cannot be managed as a conventional crop, have low biomass, and often have a short life cycle. After that, they also test on different species that have phytoremediator power, such as pelargonium, sunflower, maize and ambrosia, tilespi, and also trees like poplar and willow, because they have deep root trees. So, however, none of them possess a complete set of characteristics beneficial for phytoremediation, and they are not, in most cases, they are not promising commercial candidate.
What we can take away here is that by developing GMO, phytoremediation can be made efficient. GM crops incorporating the genes of hyperaccumulators are certainly a good idea. This solves the problem of inefficiency of hyperaccumulators. The ideal phytoaccumulators would grow rapidly, produce high amount of biomass, are tolerant and accumulate high concentrations of toxic metals, and can contain substances that prevent predation. However, there are certain concerns. The first being that it is a GMO. Therefore, releasing it into the environment without fully understanding it might lead to more harm than good to the environment, especially in terms of ecology. Secondly, since the heavy metal uptake is from the roots, the soil will only be clean at areas surrounding the roots. Nicotiana glauca, however, does not have roots that penetrate deep into the ground. In conclusion, through agrobacterium-mediated transformation, the induction and expression of wheat genes encoding phytokeratin synthase in Nicotiana glauca increased the tolerance of heavy metals such as plumbum and cadmium. Developing seedlings are 160% larger than wild types. In addition to that, seedlings grown on mining soils accumulated 2 times the concentration of heavy metals compared to wild types. These results indicated that the transformed Nicotiana glauca represents a